Hello everyone, my name is Pooja Jain. I am a faculty at the Drexel College of Medicine and I have been a member of AAI for over 5 years now, and this meeting is especially important for us because for the first time our lab has been provided with the laboratory travel award as well as graduate training award, and I'll be giving a talk in the block symposium on Sunday afternoon. So I'm very excited to be the part of this special meeting, and all the new things that I came to learn was a amazing experience for me thus far, and I look forward for next few days to be part of the meeting. The first AI meeting I attended was in San Francisco, and since then, in fact, the AI staff as well as the JI members and all the interactions I have during that meeting has really helped me to shape my idea and in fact bring lot of immunology in my research, because basically I am trained as a genetist and the person who has worked with multi-drug resistance. So attending these meetings and be part of the AI advanced course in immunology really advanced my career to bring the immunological perspective in the research that we do in the laboratory. For me, the aha moment of which kindly glued me to the field of the dendritic cell biology was when I performed these the live in vivo imaging of dendritic cell trafficking into the central nervous system, and I imaged and videographed those movements of DC across the central nervous system and to microvascular endothelial cells. I think that particular experiment I would never forget, and since then I never thought of going back to any other field or basically thinking about bringing something else other than what I stick with the importance of dendritic cell in central nervous system and neuroinflammatory diseases. I think those experiments really helped me to glue to what I am doing today. In our research, as I mentioned, focuses on understanding the dendritic cells and T cell interactions, their crosstalk and the mechanism involved in the human chronic viral infection as well as the neuroinflammatory diseases. So some of the tools that we have developed, actually we created this polychromatic antibody cocktail that helps to investigate all functional and phenotypic marker of human blood dendritic cells on the frozen, fresh or cultured PBMCs from chronically infected patients. We are also involved in developing these co-inhibitory receptor and ligand cocktail, which would allow us to investigate a number of co-inhibitory signaling on a very few cells, especially from the patient, where we have limited number of patients and limited number of patient material. So that I would think something that our lab has contributed tool wise. We have also created this new hybrid line of mice which have MSC class 1 crossbred with CD11C DTR, and that work is recently published, which allows the investigation of MSC class 1 restricted immune responses in the absence and presence of dendritic cells. So those tools are available to the community, and the uniqueness of our research which we are applying for the active and passive strategy for dendritic cell based immunotherapy. So passive strategy for the diseases such as multiple sclerosis and HIV encephalitis, and active strategies for the diseases like brain tumor and glioblastoma. So everyone in the lab, I'm very blessed to have worked with some really extraordinary hardworking people, are motivated towards contributing not only in the basic immunology but also translational aspects of the immunology, and that's something we are very hopeful to make contributions on. The AAI meetings are great forum to create networking, meet with your colleague, but also initiate new interactions and constantly follow up. So what I see that over the years the members pretty much stick to the committee. So once you meet somebody first, you were more likely to see that person again in the next meeting, even if you have not seen that person over the period of year or the meetings during the durations of one meeting to other. So the AAI meetings provides a great networking experiences to all level of researcher, whether it's a graduate student, postdoctoral fellow or scientist like us. Something that actually by coming to this meeting that I learned, which an area that we are still building on, was so much focus on the autoimmunity. For example, the great talks on the lupus which I generally never get to know lot about it. So I think my knowledge about the autoimmunity in general has been strengthened by coming to this meeting. And it's only the second day, so as the meeting go on, I expect to learn many new areas of the investigation that we generally don't pursue by ourselves. I think our contribution so far, I guess, in the field of immunology has been that we focus primarily on the human immunology and our ability to work, basically maximize the uses of the human samples, human blood material, as well as our ability to work with cryopreserved samples and clinical material from the chronically infected patients. So that is something our strength, and I think to be able to develop that strength, we are now accessing the cohort of these patients, whether it's the HTLV infected patient or HIV-SCV co-infected patients from all around the world, but we are still able to perform functional immunological investigations on that. We have also, for the first time, I guess, convincingly provided the evidence that DCs are not just a peripheral immune cells, they are very well present in the central nervous system, and their biology and function could be harnessed for the future immunotherapies. So those are the two places where my laboratory has developed its strength, and we constantly look forward building on those strengths and applying for the translational immunology. What I love most about what we do in the lab and with our research career is I've been always thought that seeing is believing. That's something that I have always loved about working with the cell biology. So that the immune cells that we work on dendritic cells, and the when we see those cells responding to inflammation in the real time and moving towards the target and performing the function, those things always give us immense pleasure. And now that our research is becoming very translational, we hope to contribute in this ever increasing field of DC based immunotherapy. So with this incentive every day going to the lab, that hopefully something we can contribute to the field that can one day help the people with diseases and malignancies. And that the thing make us going and performing our work every day as a researcher and a scientist. I think the most important thing of immunology is that each day it provides this it's like a self-stimulating field. Basically, knowing the immunology, working with the immunological problems or finding the questions, I think it makes the investigator or researcher a self-fuel to go about their research. So it's a very I would say it's a field where someone can develop passionate with and derive self motivations to work on, and that's my opinion about the immunology.